Hello children, in the last class we have studied that DNA is superior than RNA. Okay, today here in this class we are going to study about replication of DNA. Now you see here, DNA is a double helical structure. Okay, now during the process of replication, this DNA strand separate, okay, and this act as a parental strand or template. It is otherwise known as template. On this template or parental strand, this is also act as a parental strand or template strand. On this template strand, new strands are synthesized. These are the new strands which is synthesized, that is complementary to the parental strand. What do you mean by complementary? Complementary means A always pair with T, G pair with C. Okay, so the new strand is complementary to the parental strand. After the completion of the replication, the new DNA progeny or the new DNA is having one parental strand, other newly synthesized strand. This type of replication is known as semi-conservative replication. This type of replication is known as semi-conservative replication. Why it is called semi? Semi means half. Half of the parental strand is conserved in the newly synthesized, newly formed DNA. Okay, one is parental strand which is conserved and other is the newly synthesized strand. So that is why this replication is known as semi-conservative replication, and it is proposed by Watson and Crick. In the year 1953. In the same year, they made the model of the DNA double helical structure. So, there was another scientist in 1958. Two scientists whose name is Matthew Michelson and Franklin Stahl. These two scientists, they want to prove that DNA replication is semi-conservative. For that, they conducted an experiment on E. coli bacteria. They conducted an experiment in which they made a nutrient medium. That nutrient medium, heavy isotope of nitrogen is added, that is N15, in the form of ammonium chloride. In the nutrient medium, heavy isotope of nitrogen is provided in the form of ammonium chloride. In this nutrient medium, bacteria are cultured. Which bacteria? E. coli bacteria. So this is the E. coli bacteria. Okay. This is allowed to multiply for many generations. Okay. After that, actually this is not a radioactive. This is a heavy isotope. So this heavy isotope can be separated from the light by centrifugation gradient. So Michelson and Stahl, they used cesium chloride for centrifugation. Cesium chloride is used for the centrifugation. So what you have seen, for many generations, if it is grown in the heavy isotope containing, that is heavy nitrogen in this nutrient medium, so what will happen? Both the strands of the DNA get incorporated with N15. Okay, both the strands get incorporated with N15. What is the reason? Because here the nutrient medium is having heavy isotope of nitrogen. Nitrogen is present where? In the DNA. So the DNA get incorporated, the nitrogen bases get incorporated with N15. After that, another nutrient medium is taken. This nutrient medium, they added normal nitrogen, that is N14, in the form of ammonium chloride. Okay, and the same bacteria is transferred here, and it is allowed to multiply for one generation. One generation means for the multiplication of one bacteria into two cells, it will take 20 minutes. It will take 20 minutes. So it is allowed to multiply for 1 minute, means for 20 minutes, and then the DNAs are observed. It is seen that one of the strand is N15 and another strand is of N14. Again, one of the strand is N15 and another strand is N14. So here you can see N15, N14, N14, N15. So what actually happens here? The parental strand gets separated. This is the parental strand. Okay, this is the parental strand. Okay, and new strand is synthesized. This is the new strand. New strand. So it is a semi-conservative replication. So the new strand is having N14 because this nutrient medium consists of N14 nitrogen. Okay, that is why the new strand is having N14. Okay, again it is allowed to multiply in the same nutrient for 20 minutes. So what will be the result? It is seen that again this is the parental strand, N15, N14 is formed over here. Parental strand is separated. This is the parental strand N15 and this is N14. So new strand is synthesized. This new strand is having N14 and N14. Okay. Again, this is another strand, parental strand, N14 and N15 is another parental strand, N15. Okay. New strand is synthesized, that is N14 and N14. Okay, so here this is N14, N14, this is N14, N15 and this is also N14, N15. These two are N14, N14. When it is separated by density gradient, it is found that this strand is settled at the bottom, a band is formed at the bottom. Okay, a band is formed at the bottom. Why the band is formed at the bottom? Both the strands are N15 and N15. Okay, that is why the band is formed at the bottom and it is known as heavy. And here you can see, here after density gradient it has seen that the band is formed at the middle. Why the band is formed at the middle? The reason is that here the parental strand is N15 and the newly synthesized strand is N14. So we can say it is an hybrid one. It is also hybrid and it is also hybrid. So that is why it is formed, band is formed in the middle. In this case, in the third case, what you can see, two bands are formed. One is in the middle, okay, one band is formed in the middle and another band is formed at the top. So why the two bands are formed? Here you can see these two are hybrid. These two are hybrid. Why? N15 and N14. And here also you can see N14, N15. But these two are light. That is having N14 and N14, N14, N14. So that is why light band is formed here and the hybrid band is formed here. So that is why here two bands are formed. From this experiment it is proved that DNA replication is semi-conservative. How? This is the parental strand. After replication, what happened? This N15, here this is the parental strand is separated, and next parental strand is separated, N15. New strand is synthesized, N14. Why the new strand is N14? Because the nutrient medium is having N14. So another new strand is synthesized in the next parental strand. So that is also N14. So that is why it is known as a semi-conservative mode of replication. Same thing you can see here. Is it clear? So Matthew Michelson and Franklin Stahl in the year 1958, they proved that DNA replication is semi-conservative. Next we will study the process of DNA replication. Children, now we will study about the process of DNA replication. Before entering into this, we will study that enzymes involved in the process of replication. The first enzyme is helicase. Helicase. This enzyme is responsible for unwinding of DNA helix. DNA helix, unwinding, or that means opening of the helix is done by helicase enzyme. So this helicase enzyme break the hydrogen bond present between the nitrogen bases. You can see here, this is the double helical structure of DNA. It is having adenine, thymine, cytosine. Suppose here it is having thymine, adenine and cytosine pair with guanine. So double bond, triple bond. What is this bond? It is an hydrogen bond. Okay. So what is the function of helicase enzyme? Helicase enzyme breaks this hydrogen bond. It breaks the hydrogen bond and unwind the coiled structure of the DNA. Next enzyme is Topoisomerase. Topoisomerase. This enzyme will release the tension of the DNA strand by breaking the strand. What do you mean by that? You just consider this DNA as with a rope. You have seen rope, okay? Rope is also having two strands. When you stretch the two strands of the rope and when you release your hand, what will happen? Again the rope will coil. Okay. So in order to prevent that coil, you have to make a cut on the strand. So when you cut the strand of that rope, what will happen? It will turn and do not coil. Same thing what topoisomerase does. So what is the role of topoisomerase? It will cut the strand of the DNA. Okay. By cutting this strand, what will happen? It releases the tension of this strand. Okay. So that strand do not coil. So after that it forms a Y-shaped fork. That fork is known as replication fork. This is known as replication fork. Okay, so this is the role of topoisomerase. So topoisomerase releases the tension of the strand by cutting the strand and a replication fork is formed. Third is single strand stabilizing enzyme, or you can say protein. It is otherwise known as SSS protein. So what is the role of this protein? This protein stabilizes the strand. The strand is not in a stabilized condition. So the protein will bind over this region, okay, and stabilizes this strand. So this is the SSS protein. Okay. By binding on this open strand, it stabilizes the strand. And fourth one is primase. What is the role of this primase? Primase helps in the synthesis of primer. What is primer? Primer is the segment of DNA. It is a segment of RNA. It is a RNA segment. Segment of RNA. Okay. So, this RNA, small segment of RNA, which is synthesized by primase enzyme. On this primer only DNA's further nucleotides, that is DNA nucleotides, are synthesized. Okay. The next one is, fifth one is the main enzyme, that is DNA polymerase. DNA polymerase 3, which is the main enzyme for the process of replication, which is also known as DNA dependent DNA polymerase. Now listen children, this primer also synthesize, it will only synthesize in 5' to 3' direction. It will synthesize only in the 5' to 3' direction. Similarly, DNA polymerase will synthesize only in the 5' to 3' direction. Okay. So, sixth one is DNA polymerase 1. So, what is the role of this DNA polymerase 1? It will remove the primer. Primer is a segment of RNA. After the replication, after the completion of the replication, there is no need of this small segment of RNA. So, this DNA polymerase 2 will remove the primer and a gap is formed. That gap is filled by addition of the DNA nitrogen bases. Okay. Last enzyme is ligase. Ligase act as a glue. It will bind or seal the opened areas. So these are the different enzymes involved in the process of replication. Next we will study the steps in the process of replication. So the first steps in the replication is opening of helix. Opening of helix. Helix means DNA double helical structure. The subdivision is recognition of ori. Now, what do you mean by ori? Ori is known as origin of replication. Origin of replication. What is origin of replication? That means replication begins from a particular region of the DNA. So, the origin of replication is a place where replication starts. Okay. In prokaryotes, there is only one origin of replication. But in eukaryotes, there are more than one origin of replication. So, what is ori? Ori means origin of replication. This origin of replication is recognized by which enzyme? Helicase enzyme. Helicase enzyme bind at the region of the ori and they start to break the hydrogen bond which is present between the nitrogen bases. Second, under this comes topoisomerase. So what is the function of topoisomerase? It breaks the strand and it releases the tension of the strand. Third is SSS protein. What is SSS protein? Single strand stabilizing protein. So after topoisomerase, what you have seen? There is a Y-shaped fork is formed. Okay, this is a Y-shaped fork. So stabilizing proteins bind on the surface of the strand. Okay, so this is the function of stabilizing protein which stabilizes the strand. So that is known as single strand stabilizing protein. The second step is formation of primer. As you know, primer is formed by the enzyme primase. Now, you know that DNA strand is anti-parallel to each other. So, suppose this strand is in 3' to 5' direction, means this strand is in 5' to 3' direction. Primer, as you know, primer always synthesizes only in one direction, that is 5' to 3' direction. So here, this is the parental strand. We can write here as template or parental strand. On this template, primer is synthesized. Primer is a small segment of RNA. This primer is in the, again, anti-parallel to the parental strand. So this is the 5' end, 5' and 3'. So this is the primer. Okay. You have to keep it in mind one thing, that primer always forms at the third end of the parental strand. Here you see this is the parental strand, the third end is here, so the primer is formed here. What about this strand? Here there is 5' end. So here this is the 3' end. So the primer, we will consider this as a 3' end. Primer is formed over here, that is 5' to 3' direction. Why the primer is moving in 5' to 3' direction? You know that in the earlier class we have seen that in the 3' end hydroxyl group is there. So if when a new nucleotide joins with this hydroxyl group at the 5' end, this is the third carbon of the pentose sugar and this is the fifth carbon of the pentose sugar, which is joined by diester bond. That is why it is always moves in 5' to 3' direction. Okay. Now, this is known as formation of primer. Third step is synthesis of DNA. Synthesis of DNA. Now, here you see, we have seen that DNA is synthesized by DNA polymerase 3. Okay. This DNA polymerase 3 always synthesizes again in 5' to 3' direction, in the 5' to 3' direction. So here it will form a continuous strand, that is 5' and 3'. Here it forms strand, that is 5', 3'. These are new DNAs. Okay. So this DNA is formed. Now you see children, this DNA helix is opened. Okay. Now this much, this is that portion, and here this is that next strands portion. Okay. Now this strand will continuously synthesize on the basis of its complementary strand. Okay. There is no problem. But this strand will not synthesize continuously because there is no DNA. Okay. So what happened? Here, this is the third end. Again, it forms a primer over here. That is 5' to 3' direction. So this is the primer. Okay. On this primer, another segment is formed. That is 5' to 3' DNA fragment is formed. Okay. So what you can see, pieces are formed. Pieces of DNAs are formed. So these pieces are known as Okazaki fragments. Okazaki fragments. Okay. See this diagram. After the completion of this replication, two strands are formed. Here you can see a single primer with continuous DNA strand. So we can write it as a continuous strand. Continuous strand is formed. Okay. Now here you can see many primer is there. 1, 2, 3. Many primer are there. DNA fragments are there. So we can write this as a discontinuous strand. Okay. Now what is the next step? What happened? The fourth step is removal of primer and filling of gaps. Now you see here, this primer, the fourth step is removal of primer and filling of gaps. Now what happened? This primer is removed. Okay, so a gap is formed. Here also the primer is removed. Here the primer is removed. Here the primer is removed. This is done by DNA polymerase 1. Okay, that removes the primer and a gap is found. That gap is filled by DNAs. That gap is filled by the DNA nitrogen bases and a continuous strand is formed. And then these are binded with the help of ligase enzyme. Ligase enzyme acts as a glue and joins the pieces. Now you have seen that the formation of this strand is a continuous strand. It takes place faster than this strand. So this strand is known as leading strand. Leading strand, and for the formation of this strand takes little bit time. Reason is that too many primers are there, so it will take time. So that type of strand is known as lagging strand. Okay, so this is the steps in the process of replication. So we'll see once again. First is opening of the helix. Opening of the helix is due to the recognition of ori. Ori means origin of replication. On the origin of replication, helicase enzyme bind and hydrogen bonds are, they breaks the hydrogen bonds and the helix is unwinded. And after that, topoisomerase break the strand and releases the tension. That is another, one more protein is there which is known as single stranded stabilizing protein that stabilizes the strand. Okay, and then primer is added in the 5' to 3' direction, and from there continuous synthesis will takes place. Okay, one strand undergo continuously DNA synthesis takes place, but in the other strand fragments, small pieces of DNAs are formed. As this parental strand is in 5' to 3' direction, this parental strand is in 3' to 5' direction. So that fragments are known as Okazaki fragments. The last step is removal of the primer and filling of the gaps. So that primers are removed by DNA polymerase 1 and it is filled by the nitrogen bases, and then ligase enzyme joins the gaps. Okay, so this is about DNA replication. I hope you might have understood it. In the next class we will study transcription. Okay, thank you.